Good evening, students. In today's class, we are going to discuss or we are going to start our discussion for seed science and technology subject, and we'll begin with floral biology. So, before starting the today's content, I'll just discuss the course content with you or the syllabus that is prescribed by ICAR for ICAR JRF exam. These five contents comes under seed science and technology, starting with the prescribed for roles of plant hormones in seed germination and dormancy. So this we are going to study in seed physiology. Just give me a minute. The first one, that is role of plant hormones in seed germination and dormancy. So we are going to cover this part in seed physiology. And nextly, it's pollination or fertilization of in flowering plants. So this is the topic that we are going to discuss in today's class, that is floral biology. Though they have mentioned it in a very simple way that it's just the pollination and the fertilization, but it covers the broader aspects where you need to have a clear-cut understanding about the flowers, their behavior, their reproduction, and the different parts that are involved, and what are the different process or the mechanisms involved during the fertilization or during the pollination and until the seed is formed. Once the seed is formed, then we have to study the physiological aspects where we will start our discussion with germination and the hormones which regulate this germination process. So these are just interlinked. And the third one is methods in seed testing. So this is a very important topic. Why? Because if you just go through the three different year papers of previous years, so you can see that most of the questions from the seed technology is asked from this seed testing only. So make sure to attend this particular seed testing class. Okay. And fourthly, it is breeder seed, foundation, and certified seeds. Again, it's just a three different words, but we have to again cover the broader aspects here, starting with seed legislation, seed act, seed bill, seed control order, and different policies that were made by the government of India to regulate the sale of seed in India, and how this breeder seed, foundation seeds, certified seeds are produced, and what are the different organizations that are involved in the production of these different classes of seeds, and at the first place, what they mean and from where they have just been incorporated. So for that, again, we have to study the word history about the seed technology and how it started. So everything we are going to cover in this case of seed legislation. We'll talk about the historical aspects, and then we'll cover the Indian seed legislation and what are the provisions under it. So last is seed production. So again, in the seed production, we have two different types. One is varietal seed production and hybrid seed production. So much of the things from the hybrid seed production is not important for you, but again, the methods that we use for hybrid seed production, that is male sterility or CHA or and emasculation and pollination. So what are the crops that are specifically used by which of these methods? So those things are very important for you. So in that case, seed production becomes very important. So if I've just analyzed the paper of all the 3 years starting from 2020, 21 and 22. So most of the questions were from either seed testing, and it was from seed legislation, and lastly it was in the seed production. And there were questions regarding dormancy. I think one or two questions were there from dormancy. So that doesn't mean that they will follow the same pattern for next year. In the next year, they might ask you the questions from pollination, fertilization, or just by a floral biology, or also they can just ask any questions from the seed physiology. So I will just try to cover all the things in a very simple and concise way. And I want to say one thing. See, it was preparing the notes or preparing the content for seed science and technology was much hectic when it is compared to this genetics. So the main reason was lack of prescribed or the standard reference books that is available in the seed tech. See, if I just want to read a floral biology, I cannot go with a single book. I need to again do the multiple readings and take the content from the multiple books, and then I have to prepare the content. It is same for all the five different aspects, starting from seed physiology, seed testing, and seed production, and also floral biology, And lastly, it is seed marketing. So this marketing and seed health or seed pathology, these are all not there for your syllabus. Starting four things are there. So we'll stick our discussion to mainly to the starting four topics or in this particular topic. Is it not? Are the content or the outline of this particular course is clear to you all? Just raise your hand if it is yes, or you can also chat, or you can also unmute yourself and talk to me so that we can start. Okay, fine. So we'll start with floral biology. So this is the outline of today's discussion, where we'll be starting our discussions from reproduction in flowering plants, and we'll discuss about the floral types, its structure, biology, and in relation to the pollination mechanisms and effect of different environmental factors. So this is the first topic, and secondly we are going to discuss the sporogenesis, that is micro and megasporogenesis, and next to it it's gametogenesis. And then we are going to discuss the pollination, since we'll be done with the gametes that will be formed by the micro and megasporogenesis. The next step is about pollination. Once the pollination is successful, then there is the event of fertilization where the zygote will be formed, and this zygote will be developed into an embryo, and we'll discuss few aspects of how the embryo development takes place and also the factors affecting this fertilization. So once the fertilization is successful, the next step will be the seed formation and the seed maturation, and what are the different factors that are affecting this formation and maturation. And once this is done, we have two different things. One is embryo development and second one is endosperm development. So we'll just stick our discussion to these particular topics. And also I will just convey this one message. It's not like this is, you might not see any type of questions which are related to this floral biology in three papers, last 3 year papers. So again, that doesn't mean that they won't be asking anything in this year paper or next year paper. So I just try to make it simple and concise so that it should not feel that we are going out of this our syllabus. Okay, I just tried the same thing. So whatever the things I wanted to mention, I have mentioned it, and whatever things I feel that are important that can be asked in the exams, I have done it. So it's up to you whether you will just read them or whether you will just try to revise them or not. Okay. So starting with, we'll start with floral biology. So can anyone tell me like what you mean understand by the floral biology? Okay, I'll just start with the discussion. So the floral biology is just nothing but the area of ecological research that will study the evolutionary factor which have molded the behavior, structure, physiological aspects of the flowering plant. This is the meaning of floral biology. You need not to write anything from here because I will be sending the notes. But make sure like the notes whatever I prepared, it will be only for the master class students. I will be not able to share all these notes with all others. If you require these notes, either you have to go for master course or either you have to take this C technology separate course that is available in our app. Okay. So this is about floral biology. Next, there is a part in the floral biology which is called as anthropology. Can anyone tell me like what do you mean by this anthropology? I believe like some of you might have read this thing. Anthropology. Hope like spelling is clear. ANTHECOLOGY. So anthropology is nothing but the study of pollination and relation between flowers and their pollinators. So this is about anthropology and starting with floral biology. So what are the basic components of flower? If you want to know what are the basic components. Yes, pollen study, very good Archie. I didn't see your message earlier, sorry. Yeah. So what are the basic components of the flower? I hope like everyone will be knowing it. let it be, like we'll discuss it. So it's, yes, it is sepal, petal, calyx, corolla. Very good. So sepal, petal, calyx, and corolla are four different things, or they are just synonyms? Yes, others. These are the four different components that she has mentioned, or they are just synonyms? Okay, fine. They are the synonyms. And what are the other two important components? Okay, totally there are four important components of the flower. And what are the other two? Yes, very good. It's androecium and gynoecium. So these are the four basic components of a flower. Okay. So we'll just start our discussion with reproduction in flowering plants. Well, I will just come back to this flower later. We'll just start our discussion with reproduction in flowering plants. When I talk about reproduction in flowering plants, they are of mainly two types. One is sexual and another one is asexual. So sexual reproduction is by the process of pollination, where the male and female gametes will fuse with each other, which will give rise to the zygote, and this will develop into embryo and eventually to the seed. So this is how the sexual reproduction occurs in case of flowering plants. What about asexual? Can anyone tell me, like, what are the different types in the asexual reproduction, if you know it? Yes, fast. See, every time you might not able to just type the things, so better to unmute yourself and answer me. That would be the best option, because you will miss the flow if I just wait for so long for each and every simple questions. Yes, it's first one is vegetative reproduction, and second one is apomixis. Very good. And third one is clonal reproduction. So these are the three broad categories under asexual reproduction. Yes, everyone are answering it very right. It starts with vegetative reproduction, apomixis, and clonal reproduction. Yeah, pathogenesis is a type of, whether it is a type of vegetative reproduction, apomixis, or clonal reproduction. Who will answer this? Archie is mentioning about parthenogenesis. So under which category this parthenogenesis will come? Okay, how it is vegetative reproduction? See, I'll just tell you very clearly that basic difference between vegetative reproduction and apomixis. So the vegetative reproduction is by the vegetative parts of a cell, which will eventually give rise to the new plant. But when we talk of apomixis, apomixis is a process where the seed formation takes place without fertilization. So in the apomixis, again you are seeing that there are the embryo which is formed is again seed, or the material that is used for regeneration is again the seed. But in case of vegetative reproduction, the material which is used for regeneration is just a vegetative propagule that we are using. So just remember this basic difference. Most of the students will confuse with this particular difference. Okay, I hope, Archie, I'm clear. Yes. So the basic components I already mentioned. So the next thing is apomixis. We'll discuss this apomixis since it is a new topic for you all, and most of the times the people get confused in these different types of apomixis. And vegetative propagules, I don't think like there will be any confusion nor in this clonal reproduction. So we'll take this apomixis in our discussion, in today's discussion. So what is apomixis? The apomixis is derived from the Greek word which means that apo plus mixis. It is just away from mixing. So mixing between what? Mixing between male and female gametes. So it is just taking away from the mixing of female and male gametes, and seed is developed only from the female gamete. This is what apomixis means. Or the authentic definition is formation of the seed without fertilization. So this is what apomixis is. Or more clearly, without syngamy. Apomixis is a process where the seed is formed without syngamy. And there are two conditions here, or two important points to remember. One is without syngamy, and second one is whether there is a meiosis, that the process of meiosis will take place during the formation of seed in case of apomixis or not. Tell me. So whenever the embryo development takes place by the process of apomixis, is there a division of like is there a meiotic division that is taking place in the somatic cell, or from the female gamete you might take? I'm asking you like whether it will be with or without meiosis. So one thing is clear, that it is without syngamy, that is fusion of male and female gametes, right? So whether it is with or without meiosis, no. Okay, I'll just clarify this particular second point in a minute. Okay, just remember it. So just we'll start with different types of apomixis so that you will be able to clear yourself. Yes, Archie's answered it right. Like both are possible, even with meiosis event or even without meiosis, seed can be formed, and all will come under the different types of apomixis. Yes, it's right. Very good. So just tell me list out the types of apomixis that you are knowing. So I will just give you one broad classification. One is gametophytic apomixis, and second one is sporophytic apomixis. See, the meaning is very simple here. Gametophytic apomixis is because of the gamete. The spore is formed because of the gamete. Which is the gamete which is responsible for the spore production, or sorry, which is responsible for the embryo sac production, or directly the embryo formation? Spore will be always female. Whenever we talk of apomixis, it is because of only female spore, right? So if the structure in the female where the spore is giving rise to embryo sac or embryo, those type of apomixis is called as gametophytic apomixis. So then what is sporophytic apomixis? It is because of sporophytic cell, or also, or nothing but somatic cells. If the somatic cell is giving rise to embryo or the embryo sac, so then it is called as somatic cell. Clear? You want me to tell the difference between embryo and embryo sac, or you know it? S is for what? Like, should I tell you, or you already know it? Yes, well, okay, fine, good. So this is nothing but the female gametophyte. The embryo sac. Embryo is after fertilization. Even without fertilization, if a certain part of a plant which is able to regenerate itself, then it's called the embryo. Or embryo formation, again it will form eventually the seed which is capable of regeneration. That becomes the embryo once there is a successful fertilization. I hope this is clear. So this is a broad classification of the apomixis, which is gametophytic and sporophytic. So can you just tell me like any other types that you are knowing? Any other types of apomixis? Should I list it out, or you will do it for me? Have you heard about apospory, apogamy, diplospory, adventi embryoni? Yes, very good. And is there anything left? Parthenogenesis. Yes. So these are the different types of apomixis. So just tell me honestly, like, have you all understood like what is the clear-cut difference between all this process, or is there any confusion between them? Because I have seen that even while I was preparing for JRF, I was myself confused about this particularly about apogamy, apospory, and diplospory. What is the difference between these three? I was clear about apogamy, but I was not able to clear myself regarding the difference between apospory and diplospory, how they are different from each other. You people are well aware of it or not? If you are not, like, this particular diagram will make it clear for you. Where? Okay. So just remember this particular diagram where it will start with sexual reproduction in case of apomixis, or we'll just take a apomictic or asexual reproduction in both in a single chart. So in case of sexual reproduction, it is by the diploid somatic ovular tissue that will be converted into megaspore mother cell, and again this megaspore mother cell, which is of 2n, will be converted into haploid in nature, or the plody level is changed because of the meiosis. So this is again converted into 3 successful mitosis will convert this into the haploid embryo sac, which contains 7 nuclei and 8 celled structure. And once this is formed, it is called as haploid. And because of fertilization or because of the pollination, this is converted into diploid zygote and triploid endosperm. So this is the case of sexual reproduction. So when we talk of apomixis, if at the first place this diploid somatic ovular tissue directly gives rise to a somatic cell, which is again acting as a spore. It's not directly that somatic tissue is giving rise to the embryo. It is again acting as a spore. Since there has been no reduction, the diploidy level will be 2n, so that is a diploid spore. And if this is happening, it is called as apospory. But in case of diplospory, this diploid somatic ovular tissue is first converted into megaspore mother cell. So this megaspore mother cell, it is again 2n, and due to the failure of reduction into the spores, this particular megaspore mother cell directly will act as a spore. So that is why it is again diploid. If this is occurring, and this is called as diplospory. So basic difference between diplospory and apospory is, see, the megaspore mother cell is from the somatic tissue. We all know it, right? If the somatic tissue itself is acting as a spore, the process is called as apospory. If the somatic tissue is converted into or changed into a megaspore mother cell, and then due to the failure of reduction, it is directly acting as spore, then it is called as diplospory. I hope these two things are clear. So the third one is adventive embryony. So what do you understand by this adventive embryony? Yes, fast. Like, what do you understand by this adventive embryony? Have you heard about nucellus and integments? Whether these are the maternal tissue or these are the female gametophytic tissues. Yes, no sac is formed, right? So how do you define this adventitious embryony or adventive embryony? How do you define it? And what is the process involved? So this embryo is directly formed by the nucellus or integments, which are maternal in nature. So if this is taking place, then it is called as adventive embryony. So you can just continue our discussions where this diploid spore is converted into diploid embryo sac. Once this diploid embryo sac is produced, and this can be just directly converted into diploid egg, or in this diploid embryo sac, we know that there are 3 antipodal cells and 2 synergids with 1 egg cell and 2 polar nuclei. See, out of it, if this, apart from this egg cell, if other cells are giving rise to this embryo, they are called as apogamy. If in the diploid embryo sac, other than this egg cell is giving rise to this embryo, then it is called as apogamy, which is synergids or antipodals. Or if this diploid egg without fertilization or without the intervention of this male gamete or the pollen, if it is giving rise to the embryo, this particular process is called as parthenogenesis. Am I clear? Just raise your hands or just give me a confirmation if if you are clear about what I've explained till now. Okay, I think now everyone are clear. Okay, so what is the next thing to do? Even though you have clearly understood this particular chart, the best thing to do is just revise it from time to time so that you will be able to remember it. And also, like, another best thing that you can do is just explain this particular concept to your friends who have not attended it, because once you keep on explaining the things to your friends, you will be able to remember it better, because most of the students will be having the particularly doubts in this particular concept. Okay, so I've just mentioned all the things in the scripted way. You can just read these things later. Okay, so one thing I didn't mention, it was regarding parthenogenesis. So parthenogenesis, whether it was diploid or haploid, which we discussed earlier. So it was from diploid egg, and this is called as diploid parthenogenesis. If this haploid egg is giving rise to the embryo without the fertilization from the male gamete, then it's simply called as haploid parthenogenesis. So this is the difference. So both the cases are possible, where haploid parthenogenesis can also take place and diploid parthenogenesis can also take place. And these are crop specific. They are not common in all the crops. They are crop specific. Okay, fine. So this is about apospory, apogamy, diplospory, and I have just not mentioned this apogamy. Okay, I have mentioned apospory, apogamy, diplospory, adventi-embryony, and parthenogenesis. I hope this is clear. So next thing is pseudogamy. Have you heard about the term called pseudogamy? Yes, fast. So pseudogamy is nothing but pollination plus without fertilization. So the embryo is developed with pollination. The male gamete is entering to the female part or the embryo sac, but still you won't see any fertilization between male and female. Yes, very good. The students are answering it. It's nice. You have read about these things. So the pseudogamy is nothing but a process or a mechanism where the pollination will take place, but without fertilization there will be a development of the embryo. So this is what pseudogamy means. What is the other name for this pseudogamy? It is also called as false fertilization. So if I want to tell you about other types of apomixis, there are two other types. One is called as recurrent apomixis. Another one is non-recurrent apomixis. So what is recurrent apomixis? So just remember that the event of meiosis will take place in case of recurrent apomixis, and in case of non-recurrent apomixis, the embryo is directly formed from nucellus or any somatic tissue without meiosis. NR or diploid. I didn't get you. Like, it's not haploid nor diploid, or what is it? Yes. Okay. Yeah. Yeah. You can also say in terms of haploid and diploid. Yeah. Fine. So you can also say this thing in terms of meiosis, where in one case the meiosis event is occurring, and in other case the meiosis event is not occurring. Okay. Fine. So this is about pseudogamy and two different types of apomixis. So next we'll talk of flower, where we have talked about four basic components. So first thing is sepals, petals, androecium, and gynoecium. The sepals are also called as, or the collection of sepals is called as what? It is called as calyx, and the collection of petals, they are called as corolla. And what is the androecium? What do you mean by unit of androecium and unit of gynoecium? What is the unit of androecium and what is the unit of gynoecium? Can it hear you, Shweta? Akila is answering it. Yes, unit of androecium is nothing but stamens, right? Very good. And it's pistil. Yeah. So these are the four important basic components of the flower. I hope it will not drag it more. So these are the parts of the flower, and it contains four main orals, and where first two are the accessory orals, and last two are the main orals which are required for the reproduction, right? And these are the different parts of the flowers. I hope like everyone knows this. Okay, so we'll start our discussion with the pistil. Pistil is nothing but a unit of gynoecium, and which has stigma, style, and ovule. So we'll start with stigma. This is the multicellular or unicellular papillae, which are nothing but the modified epidermal cells. And again, when we talk of the surface of the stigma, it might be wet or dry, depending on the amount of secretion that is taking place. And the secretion usually contains the lipids, carbohydrates, proteins, and water. And this stigma is the main site of pollen recognition and hydration and the germination. So this is the first place where the pollen will come and drop on the pistil or a gynoecium. It's the first part. So where it acts as a site of recognition, hydration, and germination. The second part of the flower is a style. Again, we have three different types of styles based on the content inside this transmitting tissue. Can you name the different parts of or different types of the styles? If this is stigma and if this is style, what are the different types of styles? So, okay, I'll just explain this. First one is open style, second one is closed, and third one is semi-closed. Open type is nothing but the central canal will be filled with the mucilage, and it has a characteristics of many monocots and citrus. So it will be just a canal-like structure, which will be just filled with the mucilage. But when we talk of closed style, it is a solid transmitting tissue with no canal, and generally this is most commonly seen in dicot and in some monocots, such as grasses. And the semi-closed, it's just intermediate condition between open and closed. So that is observed in avocado and eucalyptus. So this is about style. So next it is about ovary. So this ovary contains one or more ovules which is covered with the integments, which is formed from which forms the micropyle, and this particular ovule is surrounded by nucellar tissue, and this is called as this integments is surrounded by the nucellus and the embryo sac. So when we talk of ovule, this is the embryo sac and this is the nucellar tissue. Outside this nucellar tissue, you will see the integments, and this completely gives us a ovary. And this is about the three important parts of the pistil, which are stigma, style, and the ovary. Next, we'll keep our discussion about ovule. So the ovule. So how can you define the ovule? So when we talk of ovary, the ovary will bear the ovule on the wall of placenta. So on the wall of placenta, you will see the ovule that is attached. If this is the placenta, on the wall of placenta, you will see the ovule that is attached to it. So this is what ovule is. So ovule is also called as megasporangium. You need not to confuse with the term megasporangium and female gametophyte. Can anyone tell me the difference between megasporangium and female gametophyte? Female gametophyte is nothing but the embryo sac. that is formed after gametogenesis, or whether it is like it's a mega gametogenesis. Yes, yes, we can also say it in terms of diploid and haploid. Yeah. Okay. So this is about the ovule, which is a part of the ovary, which is attached to the placenta. And what about nucellus? So nucellus is a central part which contains this embryo sac or the female gametophyte. Very good. So nucellus is the central part or central body which contains the embryo sac or female gametophyte. So this base of this funicle or this base of this nucellus tape, tapers to form a structure called a funicle, and which is again attached to this placenta. This funicle is again attached to this placenta, which is attached to this nucellus. Outside this nucellus, we see the outer covering, which is called as integments. And these integments actually arises from chalazal end, and in the micropylar end, there will be a short gap or minor gap. Why there is a gap in this micropylar end in this case of integments? For what purpose? So when the complete like integments are covering this nucellar tissue, you just see the small gap in case of like micropylar end. So why there is a small gap here? What is the significance of it? How does the pollen entry takes place? From which part of the embryo sac that in which part of this embryo sac the pollen entry happens? Yes, it is from the gap that is produced in this by these integments, where the pollen can take entry into this embryo sac. Yes, I hope this is clear. Okay. And once after maturation, or once after the maturation, this particular funicle will detach itself. And what is the scar that is formed in that particular region where the detachment has been taken place? What do you call that scar as? It is called as hilum. Okay. I hope all these things are clear to you. And there is some area between hilum and chalaza, and this area is called as raphe. Okay. The area between chalazal and the hilum, it is called as raphe. So this is about the ovule, integments, and the nucellus. Next, it's about integments. So there is a term called as aerial, which is just the outgrowth of this particular integment, and we have different types of structure which are related to integments in different crops, which are crop specific. You just need to just list them out and just remember them. Okay. And there can be again the outgrowth of hilum, which is called as strophiole, and this particular strophiole has a function to restrict the movement of water inside the seed. And we have a particular structure called as arbitrator, and this is again the ovular structure which is directing the growth of pollen tube, and this is a maternal or this is originated from the placenta, and this will eventually degenerate after the fertilization. So if you just want the same thing in the figure for you. Yes. So this is the obturator. I'm not able to pronounce it. Obturator. And this is the site where the pollen entry can takes place because of this particular part. Obturator. Okay. And nextly we have two different structure which is called as hypostase and epistase. So hypostase is nothing but the group of cell that you see between chalazal region and the embryo sac. So this is the hypostase which you see. These are nothing but the group of cells that are between the chalazal end and between the embryo sac. If this is the complete embryo sac and this is the chalazal end, these are the group of cells between them, and they are called as hypostase. And this will become This will thicken eventually during the maturation due to the lignification, and these are generally poor in cytoplasmic content. So this is about hypostase. But what is the use of this hypostase and what is the main function of it? So this will provide a barrier for the developing embryo sac, and it also prevents the pushing of this embryo sac downside. So this is the main function of this hypostase. And we have epistase just opposite to it, which is cells above the embryo sac and towards the micropylar region. And this will have the role to play in the pollen tube growth. Epistase. So these are the two different types that will come in the integument part, near to this integument parts, and which have some unique function. Nextly, I have also mentioned about different types of micropyle and different types of ovals based on the integuments. If you are interested to cover all these things, you can cover it. Otherwise, you can just leave it. Next, most important thing is to discuss different types of ovals. Can you just list out the different types of ovals for me? Starting with anatropous oval, orthotropous, kemphalotropous, cercinotropous, amphitropous. Is there anything left? Next one is ametropous. So these are the different types of the ovals. So from the exam point of view, what is important for you? You have to remember the examples. It might be asked in the master following, or it might be in statement wise, where they will ask like the oval will be bent at 90 degree. Type of oval which will be bent in 90 degrees, ametropous or orthotropous, and they will ask you to just correct this statement or tell which statement is correct and which is wrong. So just remember the meanings of all these type of ovals and the examples for it. I've just listed out the things, and you have a very peculiar figure for it. If you just understand all these figures, you'll be able to remember it better. So who will tell me the difference between amphitropous and kemphalotropous? So this is the kemphalotropous and this is amphitropous. Who can tell me the difference between these two? You can see some difference in this particular figure. I've also highlighted that point. Yes, fast. Yes, it is horseshoe. Yes, horseshoe in case of amphitropous. So, to explain it more clearly, the body of this embryo sac will be bent in 90 degree to the oval. If this is the oval, this due to the horseshoe shape, it will be exactly 90 degree to the base. So this is the difference between kemphalotropous and amphitropous. I hope this is clear. Here the oval or the embryo sac is not bent, it is just curved. It is not exactly the inverted, or it is not exactly in a straight line, but it is curved but not in a horseshoe shape, and this is horseshoe, and this is right angle to the base. So this is the difference. I just listed out the examples. If you get any other example, you can just make a note of it. These are the different types of ovals which will be very important for you. So make sure to remember them. Next, we have talked about pistil, and we have talked about ovary. Next, oval. So now we will just start our discussion about megasporogenesis and gametogenesis. How the embryo sac or megaspores are formed, and from which the megaspores, how the embryo sac is formed. So we'll start with megaspore mother cell. So megaspore mother cell is diploid in nature. So due to the meiosis, the event of meiosis, this megaspore mother cell, which is in where the Brody level is deployed, it will it will be giving 4 daughter cells. Out of these 4 daughter cells, 3 will degenerate, and this 1 daughter cells will be nourished by these nucellar tissues. After that, because of successive 3 mitosis, this will be converted into a embryo sac, where it contains 7 nuclei and 8 cell structure. Or I think it is 8 nucleate, 7 cell structure. Sorry, it's 8 nucleate and 7 cell structure. Clear? So this is about the process of sporogenesis and gametogenesis in case of pistil or megaspore mother cell. So in exam, you can get a question where they might ask you regarding the seeds. Number of meiosis that are required to form the 100 cysts. What will be your answer? Number of meiosis event that are required to give the 100 seeds, like 100 fertilized ovi. Consider both megasporogenesis and microsporogenesis. What will be the number of meiotic divisions that are required? Yeah, you can think about it later. I just ask about formation of 100 seed. Okay, so this is the complete structure of the embryo sac, which is also called as megagametophyte or female gametophyte. Yes, it is 125. Why it is 125? It because just we'll split it into 100 plus 25. I'll just explain why it is 100. So this is in case for female. See, when there is 100 meiosis, from the 100 meiosis, you will get 100 megaspores. Not sorry, like 400 megaspores you will get. So in each megaspores, out of 4, only one will be undergoing the meiosis, like mitosis event, and you will giving rise to embryo sac. So out of 4, one is giving rise to embryo sac. Then out of 400, only 100 will give you the complete embryo sac. So 100 embryo sacs are already there. Plus you need only 25 in case of male. I will explain it why later. Okay, when we'll just start our discussion with stamens, we can discuss it. So just I hope like you have understood why it is 100 in case of females, like why we require 100 meiotic division in case of females or female megaspores. Yes or no? If no, like I can explain it again. Yes or no? Like you want me to explain, or you already understood like how it is 100 meiotic division in case of female? Or you are just thinking like that's okay, fine, good if you understood it. So this is about the embryo sac that is formed. So this is the micropylar end and the chalazal end. Near to chalazal end, just remember there will be antipodal cells, and near to the micropylar end, you will get to see the egg cell and 2 synergids. And to the middle portion, you will get to see the polar nuclei. And I will just also mention the plody level. Just go through it. Okay. And these are the integments, outer integments, inner integments, nucellus, and this is the gap that you see here. And this blue color is nothing but the filiform apparatus, and this tissue is called as nucellar tissue. So this is again 7-celled 8 nucleate structure, which is called as embryo sac. So there are different pathways of how this megaspores or how this megasporogenesis takes place. They are monosporic, bisporic, and tetrasporic. I just left it blank because you might not require this since it is not under syllabus. So if you are very keen about to know about these things, you can just Google it or you can just see it from any source. But they are not as such important for you. So this is all about the female reproductive structure, which is pistil. So now we can start our discussion about stamens. So as we discussed earlier, stamens are nothing but the unit of androecium, and the stamen is also called as microsporophyll. So when we talk of anther can be of mainly two different types. I hope like you have already seen the structure of anther. So when we talk of stamens, it is made up of a filament and an anther. And when we talk of anther has mainly two different types, monotheca and dithecous. So where monothecous means it has only one lobe. The red marked one is called as lobe. So if it has one lobe, it is called as monothecous. If it has two lobes, it is called as dithecous. So this is how it looks in the diagram. I hope you are getting these points. If you just go through the PPT after this class, you will be able to understand it better. Okay. And this monotheca will have two sporangia. What are these sporangia? So you can see the sporangia here. Each lobe is having two sporangia. If it is one lobe will have two sporangia. If it is bilobed, and it will have four sporangia. Okay, so this is about monothecous, dithecous, and sporangia. And there is also a monosporangiate. Even it is one lobe, within the one lobe there can be a single sporangia which is called as monosporangiate. This is seen in Architopia. Example is Architopia. This is the anther types and their parts. So before going to the process of microsporogenesis, we can discuss about structure of anther. The structure of anther is mainly made up of four different layers, which starts with epidermis, endothecium, middle layers, and last one is tapetum. So these are the four main layers in the structure of the anther, which have some specific role to play, and in which the tapetum is the innermost layer and the epidermis is a single and outermost layer of this anther. So we can discuss the microsporogenesis here. So once this microspores are formed, or this pollen mother cell will eventually go into the process of meiosis, where it will form a structure called as tetrad. So this tetrad will eventually separate, which will give rise to microspores. So what is the difference between microspore and the pollen grain, if anyone knows? What is the difference between microspore and pollen grain? Yes, Jayant? I didn't hear you. Yes, after maturation, pollen is yes, it is after maturation. But what exactly? It is after cell division. Whether it is mitosis or meiosis. So this microspore will have a big, large vacuole. So nextly after maturation, this is again will be because of mitosis. It will have two different cells. One is called as vegetative cell and one is generative cell. So this is pollen Okay, just write it down here. So once it is pollen mother cell, it will give you the tetrad. So this tetrad will mature and it will give you the microspores. So these microspores again with the pollen mitosis 1, we'll get to see the vegetative cell and a generative cell within a single nucleus. So this is pollen mitosis 1. So if the pollen mitosis 2 takes place, then again you see in the generative cell you get to see two spores and single vegetative cell. This is pollen mitosis 2. So this is called as binucleate and this is called as trinucleate pollen. Yes. Okay, mitosis, you have answered it. Very good. So I have just listed the functions of these four different layers. You can just go through this list and understand what are the different functions and just remember them. Okay. Where is the figure? Yeah. I also mentioned the functions tapetum. This is very important. Make sure to read it first. Different stages. Yes. So this is the microsporogenesis, where if it is pollen mother cell, so this will undergo the meiosis, which will give you the pollen tetrad, and this tetrad with the formation of the big vacuole. So there will be a big vacuole and this small cell. After the pollen mitosis 1, it will be converted into a large vegetative cell and the small generative cell. And again from next, it is from pollen mitosis 2. These generative cells are again converted into two sperm cells. Within a generative cells, you will get to see the two sperm cells where one will go and fuse with egg and other one will go and fuse with the polar nuclei, which will eventually lead into the endosperm, the formation of endosperm. So if the pollens are released in this particular stage after pollen mitosis 1, they are called as bicellular pollen, and if it is released after 2 successful mitosis, they are called as tricellular pollen. So I have just given the how this pollen mother cell is being developed or differentiated. So it all starts from the anther, where in the anther tissues there are anther primordium, which are mainly formed of parenchyma cells. These are mainly formed of parenchymatic cells, which are called as anther primordium. And these anther primordium are converted into hypodermal cells, which will eventually become prominent than other cells within the anther. So from this hypodermal cell, there will be an increase in size, which will eventually lead into archisporial cells. So these archisporial cells are very important. So because from the archisporial cells only, we get to see the microspore production at 4 different corners of this anther. So these archisporial cells will be divided into 4 corners of this anther, and from these 4 corners, you will get to see the production of microspores. And eventually, this microspore will lead to the generation of pollen grains. I mentioned this complete process here. You can just go through the same thing. Okay. And this is the figure where it is actually showing these are the microspore mother cells, which will eventually go into the mitosis, like meiosis event, which will give you the haploid microspores, and these microspores will be converted into pollen because of pollen mitosis. That's it. And again, I was talking about the tetrad. There are again 2 different types of how the tetrad are arranged. So one is specific to monocots and one is specific to dicots. And they are not important because there are so many things that should be explained here. So just ignore it. Just know it that there are mainly 2 different types of tetrad arrangement. One is successive type and second one is simultaneous type. Just to make it complete, I just mentioned the headlines. That's it. So that if anything in by chance asked in the exam, you will be able to answer it. So in case of microsporogenesis and gametogenesis, we came till pollen grain, right? From the microspores, we came till pollen grains. So now we can discuss with detail about this pollen grain and the structure of this pollen grain. So can you tell me the important parts of the pollen grain? Or the important parts of this pollen wall in specific? Have you heard about the term intine? Yes, very good. It's intine and exine. So this exine is majorly made up of what type of compounds? Or what type of polymer you can say? Yes, very good. It's poropollen. So these are very important for your exam. It's good that you all remember it. Just make sure to revise it from time to time so that you won't get confused. So this is about pollen wall. So if you want to see the structure of this pollen grain, it generally contains intine and outer part is exine and inner part is intine of the pollen wall. And this outer part you can also see the spikes that are formed. And inside this pollen grain, you get to see the generative cells, germ pore, and also the vegetative nucleus. So why this germ pore is important? Why it is important, germ pore? Again, there is a differentiation in case of exine. It is extexine and endexine. You don't have to worry about it, but if you remember these things, it's well and good. Yes, it's for the pollen tube formation and the transfer of these sperm cells. Yes, for the release of sperm cell. Yes. Okay, nice. So this poropollenin, this is particularly absent in case of Zostera. So these are all the type of questions which by rare chance might ask in the exam. So it better that you should remember all these things. So talking about the sporopollenin, this is highly resistant material which is again made up of fatty acids, phenolics, phenol, propionoids, and carotenoids. So this is the chemical composition of sporopollen. Next about structure of this wall, pollen wall, which is exine. So again, this pollen wall in case of exine has two important layers. As you can clearly see, it's a tectum and foot layer. This is fourth layer and this is tectum. And in between this tectum and flute layer, you will just see the solid supporting tissue or supporting rods. They are called as columella. So these are the layers from exine. Okay. Out of this exine, I told you there are two parts, and this is a part of exine. So these are the parts of pollen grain. Pollen germination, you need not worry about it. And about the viability, how do you define this pollen viability? So viability is nothing but ability to live, develop, and if we talk in case of plants, it is able to ability to germinate when it is given the favorable conditions. And there are two important methods to check this pollen viability. One is NMR, and second one is contrast microscope. So what is this NMR? And it here, it's called as nuclear magnetic resonance. Let's make a note of it. I want to written it here. So they might ask you these kind of things in case of MCQs. So it is nuclear magnetic resonance. So this is a technique used to check the pollen viability. So again, if you want to store the pollen, they are very short-lived. If you want to store it, you should store it in a refrigerated conditions. So this is about the structure of pollen and different parts. So next is about dehiscence of anther. So what is this dehiscence of anther? So once the anthers get matured and pollen grains are formed, it should burst open so that the pollen grains are released and they will be carried out either by insects or by wind, so that it will reach the female part or the ovary, starting from stigma, and the process of fertilization will take place. So for that to happen, there should be a dehiscence of the anther. So while dehiscence of anther, it is the middle layers which will degenerate first, and next is tapetum, and endothecium will expand. This will eventually lead into the bursting of the anther, where there will be enzyme lysis, and by the which the which will give the expansion, and the stomium, which is a tissue that is found in the anther, will split up and pollen grains are released. So this is the process of dehiscence of anther, how it takes place. I just try to give you the overview so that I won't be just digging deep into the concepts of all floral biology and botany, because it has the least importance in case of this sea technology if we just analyze last 3 years papers. Okay. So now can you tell me the answer for how it is 125? How it is, why it is 100 plus 25? Because we have seen that in case of pollen mother cell, each microspores are giving us like each pollen mother cells are giving us we call microspores in the terms of tetrad, which will eventually mature, and individual microspores are giving the individual pollen grains, which again by the two mitosis will have the two sperm cells within it and one vegetative cells. Since total 25 pollen mother cells can give us 100 pollen grains, matured pollen grains. That is why the number is 25 here. So from 25, the meiosis, like 25 meiosis will give us 100 pollen grains. The 100 pollen grains will fuse with this 100 embryo sac that are formed, by which you will get 100 seeds. I hope this is clear. Yes or no? Okay. So this is the just the overview of whatever we have discussed about the microsporogenesis and megasporogenesis. So just try to remember the difference between this microspore and megasporogenesis. Just may have wanted to make it in a single picture. That's why I taken this. So this is about the reproduction in flowering plants and until microspore and megasporogenesis or formation of male and female gametes or till fertilization. So what is the next process after the fertilization? It's about seed development, right? So we can discuss or start our discussion about seed development in the next class. I think it will be lengthy if I just continue the seed development class now. So we'll just cover it in the next class. Is it fine? So it might bore you because since most of the things that you already know and most of the things are not that necessary, but I will just try to make it more interesting from the next class since we will just dive into the actual concepts of sea technology, which you have been studying for 4 years. We just take each and every topic and we'll discuss it very clearly. Okay, since you will feel interesting once we start discussing this seed legislation and seed testing, but know like this floral biology, since they are just conceptual and you should remember them as such, won't feel that interesting to listen for the classes for all these concepts. But the notes that I have prepared, it's worth of reading and it will be very helpful for you all. Just make sure to read it once. Okay. So as usual, next class will be on tomorrow night at the same timing. If there might be like variation in 5 to 10 minutes, I will mention it in a group. Anyhow, okay. So if anything is everything is clear, we can take a leave and we'll meet in the next class. Thanks for attending.